Hello everybody, this is D. Rajashekar, lecturer in botany, Srimati Ganganeni Kalyani Government Degree College, Vinikonda. In the previous video lectures, we have seen the cell structure of bacteria and the nutrition of bacteria. In this video lecture, we are going to learn reproduction in bacteria. What is reproduction? It is an important feature of a living organism. Every living organism wants to increase its number, its population. That is the important feature of living organisms which makes them different from non-living organisms. In bacteria, we can see various types of reproduction patterns, but however, they are all not real reproduction practices. Binary fission is the only reproduction in bacteria by which it increases its number. All other remaining practices like gene transformation, transduction, and conjugation, these are all genetical transferring processes only. Reproduction means the number must be increased, but in any of these three processes, we do not see the increase in the number of the bacterial cells. And another reproduction patterns like endospore formation, cyst formation, these are also not actually reproduction practices of bacteria, but they're all defensive mechanisms of bacteria. During adverse conditions, bacteria protect its life by these two processes. So, if you say reproduction bacteria, that is only binary fission. Binary fission, it is more or less similar to that of the mitosis of eukaryotic organisms, but with little differences. In binary fission, mother bacterial cell divides into two equal daughter cells. The duration of the time is vary from one species to another species. But in E. coli bacteria, it is 20 minutes. For every 20 minutes, E. coli bacteria divides by binary fission and gives two bacteria. Likewise, 2 to 4, 4 to 8, 8 to 16, so on. First and foremost, as we all know, bacterial cell contains a single double-stranded DNA molecule. It is called nucleoid. During binary fission, first and foremost, the DNA amount is enhanced. For that, bacterial cell carries nutrition process by which it procures many nutrients in order to promote the binary fission activity. First and foremost, there is an origin in the double-stranded DNA molecule of bacteria, where two replication forks are developed which get operated in opposite directions so that the double-stranded DNA molecule is given into two DNA strands, two DNA molecules. Later on, as the number is increased, so the bacterial cell is also a little elongated. Then the mesosome in the bacterial cell distributes the two DNA molecule towards the two extremes of the bacterial cell. Meanwhile, other cell organelles, food materials, everything is equally distributed with the help of mesosomes. Then as you see in the third diagram in the slide, plasma membrane also folds inwardly on both sides of the bacterial cell, so that these infolds of plasma membrane come closer so that the bacterial cell is divided into two equal cells. Meanwhile, the cell wall is also divided. Later on, when the two daughter cells are formed, they repair the plasma membrane as well as the cell wall. Thus, single bacterial cell divides into two equal bacterial sized cells. They are called daughter cells of the bacteria. This way, bacteria increase its number. So, this is the normal and a regular type of reproduction in bacteria. And another type is endospore formation. Endospore formation, as I told in the beginning of this lecture itself, it is not at all a reproduction process, but it is a defensive mechanism of bacteria whenever it has to face some adverse conditions like high pH, low pH, high temperature, low temperature, high salt conditions and low salt conditions. These are all adverse conditions for bacterial survival. So when these adverse conditions prevail, to protect itself, bacteria adapts this endospore formation process. Generally, gram-positive bacteria develop endospore formation. Gram-negative bacteria do not form endospores. Endospores are thick-walled resting spores. They are all mainly meant for the, enduring the unfavorable conditions. Luckily, endospores are formed by the gram-positive bacteria. We all know that gram-positive bacteria are less pathogenic than that of the gram-negative bacteria. If at all gram-negative bacteria would develop endospores, they may increase the disease severity in the host organisms. But there is only one bacteria in gram-positive, that is Bacillus anthracis, which develops endospores. These highly resistant endospores are generally present in the sheep and goats. When the human being or any other organisms take this flesh of sheep or goat, without proper cooking, without proper heating, as these endospores remain in their bodies, when we take such a food, automatically these endospores enter into our body and cause a disease called anthrax disease. That is why we are instructed to take flesh of any animal after proper cooking. And the main example for endospore forming bacteria are Bacillus species and Clostridium species. These are highly heat resistant bodies which can endure unfavorable conditions for many years. If you observe the structure of endospore, we can see there is a center core-like part which contains DNA, diaminopimelic acid, calcium ions, mucopeptides, ribosomes, cytoplasm in little amounts. So this is the major and important part of the endospore. Enclosing the core, there is a wall called core wall or germ cell wall. It is enclosed by a white color region called cortex. Cortex is enclosed by endospore coat or outer membrane, and which is in turn enclosed by exosporium. Thus, endosperm, the core region is enclosed by 4 different walls. That is why it is highly resistant to the adverse conditions. Generally, single bacterial cell develop single endospore only. It may be developed either terminally or intercalarially. First and foremost, the mother bacterial cell sends a little amount of DNA and cytoplasm, all of the cell organelles to one extreme. That extreme part pinched off from the rest of the mother cell by developing a septum. Later on, mother cell engulfs this daughter part of the same cell. Later on, it is pinched off and remains inside the mother cell. The endosperm is formed in the mother cell. Later on, when endosperm is completely matured or fully matured, the mother cell is disintegrated. Thus, endosperm is set free. Thus, released endospore endures the unfavorable conditions, and when the favorable conditions return, by dissolving the thick cell walls, it germinates into a new bacterial cell. Thus, mother cell is dead and new cell is formed. There is no increase in the number of the bacterial cells. So that is why endosperm formation in bacteria is not at all reproduction process or pattern. It is only the defensive mechanism of bacterial cell to the adverse conditions. And there are different methods, other methods we can also see in the bacteria, different reproduction patterns like budding, conidia, and cysts. Budding, in budding process, actually there are some bacteria which develop a small bud-like structure on its surface, into which little cytoplasm, DNA, and other cell organelles enter. So when this bud is completely matured, it detaches from the mother cell and develops into an independent life. So it is a reproduction process. Then conidia. Conidia is generally seen in a special bacteria called actinomycetes. They are also called ray fungus because of actually they are bacteria, but they are with little fungal characters. That is why they are called fungal bacteria. These actinomycetes species, especially streptomyces, show the formation of conidia. Conidia are round-shaped cells which are formed outside the bacterial cell like chains. They are called conidia. During favorable conditions, the disjunct are present between the conidia are dissoluted, so these conidia are set free in the environment. So on falling suitable substratum, each conidium develops into a new bacterial cell. And another type is cyst formation. It is also a defensive mechanism like endospore formation, generally seen with the Azotobacter, Myxococcus, and Chondromyces. During unfavorable conditions, cytoplasm and little amount of DNA of bacteria is enclosed by many layers, many cell walls, and they are changed into cysts. These cysts remain in the adverse conditions as it is. When they get favorable conditions, these cysts develop into new bacterial cells. So it is a type of defensive mechanism only. So far we have come across different reproduction patterns of bacteria. Among all, endosperm formation and cyst formation come under defensive mechanisms, not actually the reproduction processes. In the gene transformation, conjugation, and transduction, they are all gene transfer mechanisms, not sexual reproduction patterns. And but the main type of reproduction bacteria is binding fission, and in some bacteria we can also see budding in conidia. Thank you for your patient listening. We'll meet in another lecture with another video. Till then, bye. Thank you.